Okay, we are continuing. We have three more speech. If you let me not given any break and to continue, we are so late. Now I am introducing the Dr. Slater. He is the PhD candidate from Leiden University, from Netherlands, under the supervision of our dear colleague, Dr. Margaret de Vries. Prior, he worked as an intern in the lab of Dr. Ozaki and James Mitchell at the Harvard under the supervision of Peter Kipp. He will be starting his clinical internship in January 2025 to obtain his medical degree and looks forward to continue research as a clinician scientist. Please.
Yeah, thank you very much for the very kind introduction and to the organizing committee for hosting us here. So today I want to be talking about how to use methionine restriction to improve post-interventional vascular remodeling. And being here, of course, on the 4th protein calorie restriction workshop, I guess that most of us will know what methionine restriction is, but maybe not everyone is familiar with post-interventional vascular remodeling, although it was already introduced by Florent and by Margrethe. So let's start off with that one, which is basically the way that blood vessels adapt to the new hemodynamic situation after vascular interventions, for example, after coronary artery bypass grafting, both when veins are used or arteries are used, but it also happens after balloon angioplasty. And although these interventions are initially highly successful at revascularizing hypoxic tissue downstream of the occlusive arterial lesions, over time, as Margrethe already introduced, a lot of these vein grafts fail, leading to a high number of patients that again require re-interventions. But it's not solely a venous problem, it's actually also true for the arteries, where if we look at after arterial bypass surgery or balloon angioplasty, specifically after balloon angioplasty, again we have development of in-stent restenosis, leading to the arrival of the initial disease, but then in the new situation. And if we take a look as to why most of these vessels adapt, I think I want to take the lesson from Gökhan, saying that indeed it is initially maybe a physiologic response that then turns pathologic. So immediately post-operatively, these blood vessels are exposed to increased biomechanical forces, a lot of disturbed flow that then triggers inflammation and proliferation that is required to allow the adaptation of the blood vessel to its new environment. But over time, actually, this tends to overshoot, where we again have development of atherosclerosis, intimal hyperplasia, again leading to loss of patency, and these patients have to undergo new interventions. And the question that we ask ourselves in the lab is, how can we try to abrogate this maladaptive response that we see after surgery? And of course, I'm standing here believing that dietary restriction could be one feasible way to do that. And we specifically are interested, of course, in utilizing the benefits of dietary restriction that arise fairly quickly, and to use that in a preoperative setting that maybe will have its long-lasting effect on the vascular remodeling. So what we initially did, so we put mice on a high-fat diet for 3 weeks, then one group switched to methionine restriction. I'll come back to that, but we've heard it previously. One group remain on control. They undergo the vascular intervention, and then the mice go back to a control diet for 4 weeks. That will give us this clinically translatable strategy where patients in the ideal world would only have to do the dietary intervention preoperatively, and postoperatively they can go back to eating hamburgers. So the idea is, and this Chris already introduced, that indeed methionine restriction is quite potent in upregulating hydrogen sulfide. It has a vasoprotective effect that could potentially diminish this adverse post-interventional vascular remodeling. And this was the methionine restriction diet. So just basically no difference in macronutrients, but just solely restriction of the sulfur amino acids, methionine, and a complete deletion of cysteine from the diet. And then the first question that we ask, well, is this diet able to protect from arterial intimal hyperplasia? So here we have the experimental setup, and we used, Florent also briefly introduced this, where a focal stenosis is created in the right common carotid artery, which will induce disturbed flow, which will then lead to arterial intimal hyperplasia. But actually, when you put mice on a high-fat diet for 1 week prior to surgery, we see that we can more or less abrogate this maladaptive response after surgery. So indicating that it could be a promising strategy to reduce adverse arterial remodeling. But maybe, like I said from my introduction, arterial remodeling is not the biggest problem we face, which is actually maladaptive venous remodeling. So we asked the same question, but then in a vein graft model, where caval vein from a donor mouse is harvested and put into the arterial circulation, that then over time we will have these atherosclerotic lesions in these mice. That immediately post-operatively looks like this, where you can see indeed the maximal distension that over time will lead to maladaptive remodeling after surgery. And if we then look at 28 days after surgery and we quantify the intima to media area ratio to basically distinguish good from bad remodeling, we see that there is a significant reduction in the intima media area ratio, indicating that indeed we can also improve venous remodeling by short-term preoperative methionine restriction. But then the question is, of course, well, how does the diet do this? How does it function? And that my supervisor at the time, Peter, did some out-of-the-box thinking, and he started to look at the perivascular adipose tissue, which has actually long been overlooked in vascular biology, but is now getting more and more recognized as an endocrine organ that significantly affects vascular function. Whereas in lean patients, for example, it can have vasoprotective effects, but in obese patients where we have this dysfunctional PVAD, it can actually exacerbate cardiovascular disease. And we thought, well, maybe through dietary restriction, we get a transition from this dysfunctional PVAD into functional PVAD, which might explain the effects that we saw. So we adjusted our hypothesis and now added the PVAD component to it. And we then went back into the lab and did the surgeries, but now we did something else. And we either stripped the caval vein completely of its PVAD, or we left it intact, and both the donors as well as the recipients were placed on the same diet, thus creating 4 groups, not only the methionine and control, but also control with and without PVAD, and also methionine restriction with and without PVAD. And then when we look at 28 days, we get quite interesting results that Margrethe already briefly showed. When we now quantify the intima to media area ratio, we actually see quite an interesting thing, that when we compare the control without PVAD and the methionine restriction without PVAD, you don't see that much of a difference. But when you compare the control with PVAD and the methionine restriction with PVAD, you actually see a very significant reduction in the intima area ratio, meaning that indeed potentially there's a phenotypic switch in this PVAD, where it goes from, well, if you could say maybe bad PVAD that increases your adverse vein graft remodeling, where it then switches to having a protective effect. And that is also actually highly interesting when we look in clinical practice, where there's actually quite a big debate on how to do venous bypass surgery, and you have more or less 2 groups, one advocating, okay, either you should harvest the vessel and completely strip it of the adventitial layer of the PVAD, which is more the conventional group, or you have another group saying, no, you should leave it as much intact as possible. And interestingly, when looking at a recent clinical trial, when they compared the no-touch to the conventional techniques by just looking at the amount of occlusions, what they actually observe is that the no-touch technique is protective, but when you then look into the sub-analysis, you see that this is actually only true for the lean to moderate overweight patients, whereas if you look in the overweight patients, its protective effect is lost, indeed indicating that there might be that the PVAD might have protective effects in lean patients, but in obese patients it's actually not that good. So we were then wondering, what is then going on in this PFAT, and what explains the effects that we see. So we performed bulk RNA sequencing. We also did nuclear sequencing. I won't show that data for the sake of time. Pre-operatively, so after the diet, after 1 week of the diet, and also post-operatively to see if the changes that we observe also persist over surgery, or how this would affect. And if we then look on the transcriptome, what you might expect is that there is a very big difference between pre-operatively and post-operatively, which is what you might expect, I guess, after a surgery. But when we then look into the biggest hits that we got, we actually observe that specifically the inflammation was inhibited by the methionine restriction after surgery. When we look at toll-like receptor 4, tensin C, which is a toll-like receptor 4 ligand, or lyso-oxidase, which is also a known pro-atherosclerotic gene. So it appears that this 1 week of methionine restriction actually limits the inflammatory response that is induced by surgery. And that is also what we actually then saw when we went in vitro, when we just harvested caval vein PFAT and cultured it for 24 hours in methionine restriction medium, and we looked at the excretion of pro-inflammatory cytokines. We indeed see that this PFAT, upon being cultured in methionine restriction medium, becomes less inflammatory. And the same is actually also true when you would call bone marrow-derived macrophages in vitro. You stimulate them with LPS whilst they're on the methionine restriction diet. You actually see that they excrete significantly less pro-inflammatory cytokines. Overall then, giving us the idea that there's some immune modulatory effect of the diet after surgery. But there was also some other interesting findings that we had when we did the pathway analysis, when we actually found that there's increased thermogenesis, AMPK signaling, and PPAR alpha signaling, amongst others, which gave us the idea or the hypothesis that maybe indeed methionine restriction induces also browning, so the transition from this bad white adipose tissue phenotype to a more protective brown adipose tissue phenotype. And indeed, when looking just by qPCR, preoperatively and postoperatively, we see that indeed there's a significant increase in the amount of UCP1 expression, which is the marker for brown adipose tissue, which tends to go down, although it appears to be more upregulated at post of day 1. But interestingly, at post of day 1, when looking at the protein level, we see that actually this effect persists and even lasts until 28 days, when we stain for the amount of UCP1 positive cells that is increased, and a reduction in the adipocyte size, which is also suggestive of browning. So, like I said, I discussed that there is an inflammatory component, there is a browning component, but we were then asked, or well, we asked it ourselves, and then the reviewer came with the question, what about the link between the two of them? Are the adipocytes, do they have an effect on the macrophages, and maybe vice versa as well? So what we did, we actually cultured macrophages. We stimulated them with LPS, and simultaneously we put adipocytes on methionine restriction for 24 hours. Then we refreshed the medium and we put them together in a co-culture system. And this is just data that we got last week, so it's work that's ongoing. But it indeed appears that when these cells are co-cultured with adipocytes that have been put on methionine restriction, indeed the macrophages are less inflammatory, indeed indicating that there's a correspondence between the browning adipose tissue and the inflammatory phenotype that we saw. So to conclude for now, and then I'll just briefly highlight the work that we are currently doing. So methionine restriction protects from both venous and arterial adverse remodeling after surgery. That PFAT appears to mediate this protective effect, and well, it reduces inflammation and it induces browning, sorry for my typo, and that these phenotypes are most likely closely connected. And then what we are currently doing has been highlighted already, that adhering to a diet is quite difficult for patients. So we wanted to see if we induce browning by the beta-3 adrenergic receptor agonist, and this receptor is mainly driving the transition from white to brown adipose tissue, do we see more or less the same effect? And it's been reported for other adipose depots, but not so much for the venous perivascular adipose tissue. And indeed, we see that we can induce browning. That also appears to be there on a protein level, and we are currently investigating if the drug-induced browning of BFAT can actually also reduce vein graft disease. And the nice thing about that, so also hook us up if you want to collaborate. The really nice thing I think about this model is that you can also make a distinction whether it's actually the donor or the recipient. So you can either put the donor mice and inject it, or put it on a methionine restriction diet, and then see whether it's solely the small vein graft itself, or you also need the organism around it to mediate the protective effects. So with that, I would like to conclude and especially thank Jay and Dr. Ozaki for hosting me back in the days already, and my supervisor Margreet for hosting me now in her lab. So with that, I think thank you very much.
Thank you very much. I think so. Everybody is tired, but I want to change my place with Margaret. Margaret, will you chair this session? Continue.
So, let's wrap up the next section, and it's my great honor to introduce to you Professor Dr. Zafer, like mentor for a long time, and he serves as the John A. Manick Chair of Surgery and Executive Vice Chair in the Department of Surgery at Brigham. He's earned his undergraduate and medical degree from Duke University, and he still watches all the games. He stayed as a deacon at Deaconess and Harvard Surgical Services, and went on to complete a fellowship in vascular surgery at the University of Michigan. And he serves as a professor and chief of the North Florida South Georgia Community Health System at the University of Florida. He is a fantastic surgeon, and but he also does a lot of preclinical research, which is, I think, a very unique combination. And he's also has a lot of teaching contributions. He has led NIH and AHA funded vascular research for almost 2.5 decades, and he has spearheaded investigator-initiated as well as industry translational clinical studies and device trials, and yeah, it's a fantastic call.
Thank you, Marguerite, and this is going to be quite a shift of gears here. There's no way I can get up here and talk about all the fantastic science that's been discussed, but I am physiologically old now, and I've seen many young faculty and young trainees in the audience, and my talk is really centered at you, and I'm going to kind of weave in a fishing hobby I have. I love to fish. I was born and raised in Florida, and then I want to kind of tell you what it's like as a clinician scientist and some of the lessons I've learned over my career, and hopefully there'll be some little pearls of wisdom. And I'm going to kind of start off telling you what it's like to set up a lab. I'm going to tell you what I went through as a sort of a mid-career crisis and a move, and some of the things that I learned related to the science that I was studying. I'm going to very briefly touch on a new field that was introduced to me by Jay. And then I'm going to talk a lot about translation, how important it is that we think about the end game of how we can help our patients. I'm going to weave in a lot of pictures that are meaningful to me and tell you through the eyes of an angler, which is basically in English, as a fisherman. First, there are many different types of fishing. One can do ice fishing, cast net fishing, etc. And for those different types of fishings, one can do health services research, one can look at big databases, one can look at fundamental research, one can look at clinical trials. One of the most important things is early career, learn from master fishermen. Of course, I have my parents here, but I'm also showing mentors I've had throughout my career, and certainly people that were partners like Jay Mitchell were also my master fishing guides that taught me much about fishing. Focus on a core fishing ground. This is where I fished. Marguerite has been here. This is in the northern part of Florida. I really know these waters well. And as a clinician scientist, you need to really have a brand, something you really get to know well. And my brand, I was fascinated how blood vessels would change in response to physical forces. And everything I did throughout my career, whether it's working with a small molecule with industry, working with dietary interventions, etc., all tied back to that core business. There's a lot of fancy equipment you can buy, especially early career, and I see people throw money away on fancy machines that are very quickly outdated. You can really get a lot from just a very simple setup in a lab and a lot of hard work. And here's a tournament where the winners actually fished out of a little canoe rather than a regular fishing boat. Early career, I suggest working under the shadow of a mentor. That person already has a set up lab. Anytime a lab is started, new equipment is released. Look for these yard sales. Try to strategize to develop very economical models. If you go into a primate model for the rest of your life, you're going to be writing grants to ensure that you have enough money to do that. We are clinicians frequently, and we have access to human tissues. And if you can avoid animal models, use the human tissues that are discarded in the operating room. And then finally, collaborate. Not infrequently, someone down the hall will be doing an experiment. They'll have an experimental intervention, and maybe you're interested in the microbiome, and you could just tag onto those experiments and not have to reinvent everything yourself. Fishing is a very all-consuming hobby, and you got to make sure your loved ones, your co-workers are okay with you fishing. It will really take up a lot of weekends. We all have jobs during the week. On the weekends, you'll find yourself having to write grants, review papers, etc. It can also be an immersive structure that can overconsume you, so you certainly need work-life balance. And you have to kind of know your teams that you work with clinically, and they need to appreciate it, that while you may not take as much call, you're working on something that will help the trainees and the overall clinical practice. You may have to scale back. I never really did endovascular interventions, and that gave me extra time for research. Weekends can be quite productive. Your family will never complain that you're leaving too early on a Saturday morning, but if you go late in the weekdays and the weekend nights, they will complain, and they should. So you really got to learn to be very efficient in the operating room, in the clinic, and on the weekends. And then travel to an occasional fishing show. I really found that I learned a lot going to hear other scientists like at a meeting like this. The social networking is very important. It's a skill sport, but there's a lot of luck involved. And I show this picture because of those that know Anthony Lee know he is the worst fisher in the world, yet he actually caught the biggest fish in one of our tournaments. You got to be prepared for failures. I wrote 7 grants my first year, and none of them actually worked and got funded. It takes a lot of tenacity. You have to have a fire in your belly to throw that rod out many, many times before you'll ever catch your first fish. And it's very important you keep throwing that rod out and not get frustrated early career. Having a good team. You got to really choose your boat mates carefully. And this is one of the things that drove me to move to Boston. Really, I enjoyed immersing myself with smart people. I felt like it sort of rubbed off, and I'm not that smart a person, but when I come to a meeting like this, I see all the brilliant minds that come in, and I will learn things. Your lab team needs to be the appropriate scale. Sometimes my team got so big, I couldn't remember what everybody was doing. I do feel like I really got to know people at social events and when I traveled with them. You get to talk about their life outside the hospital. They feel less threatened than in a lab meeting. And so they're more likely to tell you about an idea they have, troubles they're having, etc. We really need to learn how to embrace diversity. I think people at different career stages, coming from different cultures, different work styles, actually in the right chemistry can work together and be truly synergistic. And then you have to be structured. And many times I've had people that didn't have language skills, and I'd have to write everything down, and I found that kind of written resource or putting everything on a big board helped people understand what our big lab plan was. Have a central rock. So right here you see Dr. Ming Tao. She was with me for almost 2 decades. We worked side by side at University of Florida, and even though postdocs and predocs would come and go, having that central person in the lab gave us a lot of continuity and develop a partnership with someone that knows you well, that can go through the ebbs and flows of funding cycles, but new trainees coming in and out. You have to have good bait, and whenever you're fishing, that bait really matters. But whenever you're writing a grant, you really have to attack a meaningful problem. You want to say, hey, I have a very unique tool. I can measure something that very few people in the world can measure. Make sure your hypothesis is truly testable with the things you're proposing. And then one of the most important things, we all hope our experiments are going to work. It's going to prove something. But what are you going to learn if there's no difference at all between your two groups? Make sure you write your grants in a way that no matter what the experimental outcomes, there's going to be a win for everyone. Spend but never lie. And I always like to show my mentor, Dr. Moldauer here. He was at University of Florida, and he was an English major. And he was very passionate about his science. He was a very good writer. And he would write hyperbole. He would talk about how excited he was about his science, and it came across in the application. But you really have to make sure that you're being truthful. You're just showing all your enthusiasm about your work. Have multiple sponsors. You know, if you have a good fishing team, these are all like sponsors. In the United States, you get $100 if you use this type of lure, etc. In the United States, I'm sure it's just like Turkey. You want to kind of have partnerships with industry, the federal government. You want to have foundations and private donors, etc., that can help support your lab work, even if a grant doesn't get funded, etc. We call it having a diverse portfolio, so you have a lot of different funding streamlines. Fish fertile waters, but don't be afraid to move. So things were going pretty well at University of Florida, but I could tell we're about to have a rough time. And also I'd kind of come to a dead end scientifically in many ways, and thus I felt I was going to need a new move. I'd come to some certain projects and I just needed to stop them. I liked surrounding myself with smart people, and I also was learning mid-career that it's okay to say no. I do not want to be chief of staff at the hospital. I want to spend more time doing research, etc. And I do think you have to really evolve to a laser focus if you want to elevate yourself to be one of the top people in a field, and you can't be trying to do everything at once mid-career. So the vascular landscape changed. We were very focused on this kind of cycle, and I tested a compound with a company and went through a lot of experimental work basically based on this pathway. And the long and short of it is, we thought this compound would work to really cure animal hyperplasia, and it was a complete failure. The traditional mechanistic approaches to science weren't working. We realized that science was much more complicated than it would seem just focusing on a single target. You saw some principal component analysis, etc. And really, trauma after vascular surgery mattered more than any of my little compound mattered. So we went through this E2F decoy trial, a big clinical trial that also failed, and I had a midlife crisis. I threw our dog in a little basket. This is my child, and we moved back up to Boston. And then going outside my usual fishing ground really made me comfortable and learn new things. I went away, breaking out of this kind of paradigm to the dietary restriction that we heard about today. And this was the focus of my research for quite a while, almost a decade. And it was a completely different area that led to many different papers, really kind of transferring things. I was stealing things that Jay was looking at and brought him into vascular biology. And it was a win for him, and it was a win for our team too. Collaboration can really help. We really need collaborators like Jay. Shared research trainees can help facilitate those collaborations. You want to document that you can really collaborate by having joint papers. And I'm really about, to me, 1 in 10 people that I started a relationship actually get to the level where I'm writing a paper. So you early have to go out and get to know people, but you have to match at some point with regards to your work style and what your interests are. Landing a big fish takes a team effort, and this is where I look at some of the people in the audience, like Dr. Hine here and his big cell papers, etc. When you catch a big fish, it's never just you by yourself. And whenever you have these big papers, you'll see a lot of authors, because in the modern age, it's going to take clinician scientists, basic scientists, those that analyze the data, those that are good writers to actually make something like this happen. Remember, you do have a real job. You have to make sure that you carry your weight in the clinical call duties, etc. Make sure you keep your partners informed on what you're doing. Make sure that your niche in the clinical practice might have to be quite limited. And also realize, too, that whenever you do administrative service, it can really take away from what you do in the laboratory. So be careful about taking on administrative roles. Don't waste time on fish that you can't eat. And by this, I mean, we don't do research on things just out of curiosity. We should focus on areas of clinical need. And we need to pay attention to the intellectual property issues. Again, Chris and I were talking about this. This can really take the things we discover in the lab to the bedside. We have to explore practical translatable approaches. And this is something we should all talk about at dinner, is that, yes, the science is very original, but how can it really translate when it's hard to say that there's money to be made just helping patients at this point, especially in a fee-for-service area where the more complications you have, you actually may make more money as a clinician. So we always fought to go to translation, and one of my clinical goals was, as a scientist, I wanted to have something go to clinical trial. And we actually had 2 small trials that we've done this, not even counting the one that Alvin and his team did in Lausanne, where we studied dietary interventions in people undergoing vascular surgery to look at the safety and feasibility. And just like many of you in the audience, I kind of had this sort of paradigm, and I was very proud of it, and I got it to this stage. But losing a fishing buddy devastates, and I can't get emotional now. I've already told people I can't do it. But really, losing Jay was devastating to me personally and professionally. We had done everything. We lived in the same neighborhood. Our kids went to the same school, etc. And many of you kindly came to Boston to celebrate Jay's life. I will never forget that moment with special friends. We had a pandemic. I've evolved personally. And, many of my research commitments have kind of gone away to other administrative roles. But this is my final message, and that one of the most important things we could do is take a kid fishing. And by that, I mean there are many young people in the audience. And I know I'm never going to get this to the point where there's a small molecule that's a dietary restriction mimetic, etc. But so many people that have come and gone through the lab, and I see them carrying the field forward. And this is very rewarding for me personally. This is probably my most important thing. People I've touched, people that have touched me at the same time, they are just as important as collaborators. I'll show here, this is Steph Curry, who sat at the end of our dock fishing. He became an MVP of the NBA. This is someone that became a politician in in the United States. He actually is someone we won't talk about at dinner. But in reality, when you take a kid fishing, this is the easiest money you're going to get. These are the early grants for early career investigators. They bring in fresh ideas, skill, energy, and you're repaying the cycle of mentorship. In reality, they're a key research partner. It's a two-way mentorship stream. And you'll see enormous pride in seeing them move the field forward. So in summary, everything you wanted to know as a clinician scientist can be gained by fishing in the sea that's right outside our door here. You need to develop a core business. You need to have tenacity. It's a team science. And often we transfer knowledge from one field, like dietary restriction, into my vascular biology world. We need to leverage our unique skills, our knowledge of clinical context if you're a physician, and identify the true care gaps, and know that we have access to patient data. We have access to patient tissues. We know how to do clinical trials. And then finally, very important that we translate, that we really do need to push from the bedside to the patient to really move care forward. Thank you very much.
Thank you very much, Keith. Thank you, Keith, for this wonderful life lesson. And I think we'll have Nothing to add to this.
So, thank you so much. I know everybody's very tired, and I just want to give a couple minutes of chance to any of you who wants to say anything. Either in Turkish, English, different language. We're happy to translate. We don't want to take this chance away. And I want to introduce Kristen here also. Thank you so much for coming. And we all talked about translation. And after this great talk, I cannot be anything but speechless. Thank you so much to share all these things with all of us. But before we fully conclude, is there any question, comments? What do you think about things? Like anything?
Sorry, one more slide. So, and one of the things you've said repeatedly is that someone's here with us, and so I'm going to put this up so he can hear the discussion. So.
So I am now a practicing vascular surgeon outside of New York City. I was one of the future trainees, so both literally and figuratively. He has taken me fishing, and he is mine for today, and I've had so much success in my career currently. So, thank you. But I thought that Dr. Cantürk had a really great quote in one of his lectures today from Hippocrates about the gut is the source of all disease. And I think today all of these talks really kind of were representative of that, from bariatrics to exercise tolerance to cancer treatment and ways to optimize that. So I think obviously there are many ways we can go with this, with many different specialties that we could spend hours talking about.
I think Christine's an interesting person to have as a part of this discussion because she is an athlete. And I was always fascinated first talking to Jay about how athletes in reality, when they do carbohydrate loading in reality, they may be restricting their protein, etc. And so it's there's so many other sort of parallels beyond the health span, along from beyond preparing someone for an acute elective surgery, etc. But even spilling over, you know, we one of the funnest meetings I ever went to with Jay is the U.S. military pulled us out and said, you know, if we're going to put a soldier in harm's way, if they're going to be hurt, is there a way that they could lessen the chance they develop a serious illness, or maybe they would recover faster, etc. So, you know, rather than eating a steak before going into battle, someone ought to have pasta and restrict the number of calories in total. But, you know, the span of beyond the conversations today spill out to many other aspects of our society beyond just simple surgery and health span.
So, yeah, so great talks. And just an observation, we flipped from like amazing, you know, preclinical studies, animal models where we got great control of the diet and these mechanisms, and I guess the clinical people in the room kind of think, yeah, but does it happen in humans? And then we flipped to a human study when people don't quite do the diets and we have adherence. There must be some line in between where we do good controlled metabolic studies in humans where we're manipulating the diet in mimicking the animal studies, but in, you know, obviously it would have to be short term unless you're not up in the, you know, the calorie dome. But there seems to be this middle ground that I feel we miss. I mean, 'cause what we're seeing in the mice is obviously giving us a big signal, but it is going to be different, isn't it? There's different metabolism, etc. So I don't know what people. I mean, they're not easy studies to do, but I just think that's where we go. And then it's about how do you get people to do it. But if we don't quite know what people should be doing, I think that's tricky.
It's the middle ground is not easy to find, but I think you can only find when you really combine the forces. So every country has its advantages, disadvantages. Every group. That's why we are here. So when you we were discussing with Dr. Alania that, you know, when you ask someone not to drink alcohol at all for 20 days might be say, I'm not going to attend this trial. I'm not going to be a guinea pig in this kind of things. I'm okay. But versus in here, it's not even a consideration for many people. You can do that trial here. There's many different things that you cannot do here, but you can do in Germany. Versus, you have to communicate, I think, and find the middle ground and say this is doable actually for human. And then, all the things that we discuss is huge problems. The cancer patients, the longevity, diabetes, obesity, all these things. We have to find the middle ground. So.
Amazing results in the animal studies when you go into the clinic and then people don't quite do it right and you go, oh, it doesn't work.
Exactly,
So
exactly.
Because it doesn't work in humans, it isn't in humans. So we have that sort of mechanistic.
Right, Dr. Lotto and I take care of patients that are not known for making good decisions, and they are not known for their discipline. These are people that overindulge with nicotine and eating, etc., don't manage their diabetes, aren't attended to their kidney failure, etc. But we've done 2 small trials, the initial one inpatient, then one outpatient. And actually, when someone is about to get surgery, in the South, we have this phrase, they come to Jesus. They actually, especially if they have a spouse, they say, okay, you're going to do it. And one of Jay's biggest contribution is you can do this very short term and have a metabolic effect. If you calorically restrict for 4 or 5 days, you're going to feel very different. And so we did find compliance is actually reasonably high, even in the vascular surgery patient. And of course, I'm impressed when someone gets cancer, often they say, okay, I do want to do everything. I know it won't fit for everyone, and this is the attraction of maybe a pharmacologic approach. But I do think there are a few times in someone's life where they really might get serious about this, especially if they have the social support, etc.
So I have a question to the other researchers, because I really enjoyed all the presentations, but most of us either used males or females. Do you think there's a different response to the dietary restriction strategies in males and females? And what is it?
Claudia, I think Michelle had some answers already.
So, thank you for this question. It's actually crucial. So, in the lab, the vast majority of groups working on calorie restriction and cancer historically used and still use female mice. We recently conducted an experiment using the same tumor cell line injected in the same mouse strain, but in females versus males. And in the case of watching-only fasting, we only observed the responses in females. In cancer patients, I cannot say, because we, as previously shown, we observed very good and long-lasting responses in both male and female patients. But all these tumor responses were obtained by combining fasting mimicking diet with standard treatment. So I don't know. But in mice, there is, at least in the models, in the biopsy mice, we used a clear difference between mice and females.
I can add from the field of nephrology, it's the other way around. So male mice are much more susceptible for AKI, and we know that, and we use caloric restriction only in male mice because we know that female mice, we need much higher numbers, and it's basically, we found out due to an to 20 HET, so one of the downstream targets is in cytochrome C derivate, and it's only present in male mice. And so there we had kind of a link. With regard to humans, we don't see that, males are more susceptible for AKI in humans. So there's also a big gap between rodents and humans.
Exactly, and maybe Chris.
Thank you. So we've actually done, I didn't present it today, we've done a couple male and female combined studies. So in our hands, both male and female mice under caloric restriction gain the benefits. You put them on a 35 to 40% daily caloric restriction, starting at about 5.5 months of age, human equivalent about 35 years old, and they both had about a 25% increase in median lifespan. However, when we tried intermittent fasting, we only saw benefits in the male mice because the female mice on their fed days actually ate like crazy and overconsumed. So I think there is sometimes a disconnect between a fasting response in of itself and then caloric restriction. And one of, I don't want to say a downfall, but female mice aren't a great model for women, while male mice might be a good model for men. In the sense of, there aren't many mammals that undergo menopause. Humans are one. There's some whale species that do it as well. Mice don't. And so when you have this kind of, female mice can kind of reproduce until later life, but female women, so the hormones are very different, and the drive of using resources for reproduction versus using resources for survival are very different between female mice and male mice. But I think it's very important for any researchers going forward, you include both sexes, because you're going to get your answers better doing that, and translation to humans better doing that than just choosing one.
Yeah, I was going to comment equally on the menopause, but I'll make it even more complicated for you. So we're not only using either male or female, but I think a bigger issue, we're using mouse strains that are inbred for generations, and there is no genetic sort of diversity in there. So we're looking at it's not representative of what humans are, period, and particularly the B6 mice are, I think, by a lot of people in the aging field, sort of now viewed as the worst model to pick, but we keep going back to it because they're well characterized.
So much. Any Turkish comment? I know you have an idea. You came, there are many students here came just for this from all over Turkey, from Adana, from Hatay, from many different places. So you were curious, and such an incredible curiosity. So first of all, congratulations for this curiosity, which will lead to fishing and probably feed. So I really wonder what you think. There's something in your brain. I know that maybe you're thinking, or you just hesitate to speak, but don't hesitate to speak. This is time to speak. This is always, you're valuable, your ideas are valuable, even more valuable than mine. So if I speak, you should speak. I really want to hear at least one person say, this is what I think about today. I'm not going to count on you, there will be one.
Actually, I want to thank you for today because I had the chance with meeting with Dr. Mitchell before. I'm a medical graduate from Kocaeli University at 2022, and I had a very, very high chance to meet with him at 2019. I participated in a research project here in Kocaeli University, and I had the chance to present it at Harvard PHN School of Public Health. And we met there with Dr. Mitchell, and he was a great person, and he was really, really inspired us for science, actually, like you, Dr. Breck also. And he was here in Kocaeli after we were in Harvard, and I know that he was really busy. He has a very project in his mind, but he remembered my work on research, and he asked that, what are you doing right now? Like, I was working on breast cancer cells, and my research project was not came to an end when I was presenting it. And he was asking me that, what did you do with those cells, and what did you do with the experiment? And I was feeling really honored because he was a great scientist, and he remembered my project after all those months. So he was a person that trying to take kids to fishing, and he was trying to teach us to fish. So I'm really happy to talk on behalf of him today, and I'm really honored to have you all here in Kocaeli University. I'm proud of my university today. So thank you all for coming. I just wanted to thank you all And I'm really grateful for today. Thank you.
We will continue to Jay's spirit. This is not the end, this is just the beginning. I will get your word and I will leave the stage to that. Please introduce yourself.
Okay. Hi everyone, I'm Elif and I'm a medical student here in Kocaeli University. This is my 5th year here, and I just wanted to thank you because this is the chance that I can talk, and I wanted to thank you because I think that's a great chance for us to experience the scientific presentations and the scientific environment that we are going to be in the future. And also, I want to add that before I started the medical faculty, I was confused to choose a medical faculty or to choose a biological field. But I met some scientists and they told me about you, sir, and they told me about that you can still pursue a preclinical field after you graduate from medical school like Dr. Furkan Burak. And I was, okay, then I can, maybe it's gonna be 6 years, but I still can pursue a field that I want to, and it's going to be a lot of options in front of me when I graduate from medical school. I'm still not sure if I want to be a clinician, but it's still great to see all of the options here, and I just want to add this. Thank you, and thank you for the presentation.
Thank you very much. Today is very excellent day. I am very pleased to host you here. But I would like to tell something about the science. Science is not just fishing. Sometimes curiosity, sometimes jogging, but never quit. Today, I made an opening speech for Turkish, our students meeting in Turkish Kocaeli University students research groups meeting. Almost 700 students participated. I told them being curious is very important and then never quit. As you know, in social science, there is no symmetry. Social science is asymmetric because time is asymmetric, for place asymmetric. What I mean, whenever you are talking about democracy, it is not the 20th century's democracy is similar to 15th century democracy. And democracy is not similar between the countries. It is not same for United States. It is not for same for Turkey. But science we are talking about now has a symmetry. But life sciences is not have symmetry. It changes with the race. It changes with the sexual differences. There is a many diversity, so we have too much way to go. And I believe that these guys, on your mentorship, on our mentorship, will make better studies. Whenever we are here or other side, we will watch their success with happiness. I hope so. These guys and the guys all around the world who are interested that scientific works will make the world better place. Anyway, we are trying to the world as a desperate place because of changing the environment, etc. You know. But I hope so. They will create a better world for us Thank you very much being here. Thank you everyone. Until this late time, they are with us. We did not plan to until 6 o'clock, but time is 6 o'clock. Thank you very much for everyone. And now, if you let me, if you want to say anything more, would you please, you can say. But if you do not, we will give you a participation certificate to you, appreciation, and then we will give you some gift for remembering us, okay? And then we will take a picture at the here. Everyone can participate us with the picture. But first, I will invite two colleagues to give their certificate and the gifts. First, I am inviting the Charlotte. You are a lucky one, as you know. Remember us, and I hope you would like. Okay, you will enjoy. Okay, next one. Chris Hine. Chris. Anyway, thank you very much you for helping us to plan this meeting. Thank you. This is the small remember. All right. Gladio. Thank you. Being here. Keith. Keith, thank you very much for everything, what you did until today for the science. Thank you very much.
Oh, thank you.
Thank you very much. And now, Christian. Buna gerek yok herhalde arkadaşlar. Sesiniz. Evet.
Şu kapatalım istersen.
Thank you very much. And Felix. Thank you. Take home our best wishes. Flora. Thank you very much for coming here. Furkan. Margaret.
So,
Yes. Yes. You are very clever. Thank you very much.
Maria, we're going to transfer you with this stuff.
Michelle? Thank you. And Sebastian. Long trip. Thank you very much. Thank you. Yes. Tish? Tish? Hi. Thank you very much. And now, if we can give our ladies who contribute this meeting, come here, please. Birer tane hediye verelim arkadaşlar. İki hanım arkadaşımıza da. Evet arkadaşlar siz de gelin. Hep beraber fotoğraf çekineceğiz. Bütün arkadaşlarımız bu tarafa gelsin. Arkadaşlarımız hepimiz buraya gelelim.
Everybody else?
Ön tarafta durabilir gençler.
Gençler katılabilirler. Bulamıyorum ki. Sen de buraya geldin.
I will do that. Arkadaşlar, çıkın bir kısmınız yukarıya. Bakın biz böyle dizimizin üzerine durduk. Bizim kızlar siz de katılın.